Welcome back to Educator Fresh, and today we are going to be talking about the worked example assessment objective 2. In the previous assessment objective, we talked about the command words that we use when we are talking about when we are assessing assessment objective 2. Some of those words include analysis, where we break down data about crops, photosynthetic data, we identify the causes and effects for a biological explanation and what you must avoid. We talked about words like examine. We talked about words like account for, evaluate, where in evaluation we are going to judge an agricultural strategy and its effectiveness using evidence that has been provided. In this video, we are going to do as examples of this assessment objective and how we can answer the questions that are going to be in this particular assessment objective. Now, here's our worked example, and I have titled it The Maize versus Bean in a Drought Scenario. Now, let's read together. Smallholder farmers in eastern Uganda find maize consistently outperforms beans during the dry season, despite similar fertilizer input. Pause there. That phrase right there, despite similar fertilizer input, has a very big meaning when you put it in context, because this is going to mean a lot of things. And I want you to learn to notice phrases like this. And you ask yourself, why is it there? Why have they put this particular phrase in this scenario? Because in this case, if fertilizer, that means nutrient availability, is the same for both crops, then nutrients cannot be the explanation for the difference in yield. The exam is explicitly closing off the wrong answer for you. It is telling you, do not write about nitrogen or phosphorus. The gap is physiological. It's not nutritional. That is clue number one. And guess what? It is sitting right there in the first sentence. So train your mind to look out for those clues when you're reading the scenario. Let's continue. Research shows that both grow under controlled light intensity and temperature, measuring net photosynthetic rate. Leaf cross-sections show that maize has a crans anatomy. Pay attention to that. Now, a crans anatomy is where we have bundle sheath plus mesophyll chloroplasts, while beans have a uniform mesophyll cell arrangement only. This is our second clue, the crans anatomy. If you remember in topic 2, the crans anatomy is the defining structure of C4 plants. It is the arrangement where bundle sheath cells, which are large cells wrapped around the vesicular tissue, sit along the regular mesophyll cells. Both contain chloroplasts, but they have different jobs. Beans have uniform mesophyll cells only. Now, this is telling us that beans are standard C3 plants. Immediately from the leaf structure alone, we know that maize is a C4 plant and that beans are a C3 plant. Do you see it? Now, this single fact is going to drive most of the answers that we are going to write in this particular scenario. The third clue, let's talk about it. Now, this clue is designed in the experimental design itself. They are testing for controlled light. Remember, whenever we are doing an experiment, we keep certain factors constant and we variate others, we change others to see how they are going to affect the rate of photosynthesis in a plant. So in this case, we have controlled light intensity and temperature. The question you have to ask yourself is, why would researchers specifically vary temperature if temperature was not expected to matter? Because we know that high temperatures plus light intensity is exactly the condition where C4s have a higher advantage over C3s. If you remember topic 2. Now, the experimental design is already hinting at the answers you're supposed to be writing. Let us look at the data on the right. Table 1. We have photosynthetic rate that is measured at different light and temperature combinations. For example, we have at 300 micromoles of light and 20 degrees, which is a relatively mild or light condition. Bean measures 8.5 and maize measures 9.1. Notice that these numbers are close. They are not identical, but they are close. One is 8.5 and the other one has a difference of 0.6. Now, at this mild condition, C3 and C4 pathways are performing fairly the same. Let's continue. When you go to 700 micromoles of light and 30 degrees, that means we have brighter light and we have warmer temperatures. Bean rises to 10.2 and maize jumps to 18.4. Maize has nearly doubled the rate of beans. Pay attention to that. Let's go to 700 micromoles of light and 38 degrees. It is still hotter. Watch what happens. Beans are going to fall from 10.2 and they are going to go to 6.1. It is not plateauing. It is going down. It is collapsing. It is dropping below even its own performance when the temperatures were cooler and dimmer. Maize, meanwhile, is going to go from 18.4 to 17.9, which is almost a plateau. It's almost flat. It's going to have a tiny dip, but it's not a collapse. Now, that contrast tells us something, that beans' rate of photosynthesis is falling off a cliff at a high temperature, while maize barely notices it. This is the single most important pattern in this entire scenario. And I want you to train yourself to see these patterns in every scenario of assessment objective 2, because they are going to be there. The emphasis of these tests, of these exams, is going to be for your ability to pick out patterns as they occur in different processes. Now, every task we are about to work through is going to come back to explaining exactly why that difference happens. Why when temperatures increase, beans fall off, and why maize remains almost the same. So, before we move on, I want you to fix that pattern in your mind. At 38 degrees Celsius, beans dropped to 6.1, maize holds at a high 17.9. You got it? Now, let's build our first answer around it. Now, our question is, analyze the data and the leaf structure to account for the differences in photosynthetic performance. Ignore the marks. Don't think about the marks a lot. What we want to ask ourselves is, what does this question want from me? So we are going to decode what this question is actually asking, because it is step 1 of our 5-step process. If you've not watched the first video, go back and watch it. We want to apply those 5 steps directly so that we come to an answer. So now, the question is telling you, it wants you to use table 1 together with the leaf cross-section, not one or the other, but both. It wants the biological mechanism behind the pattern, not just a description of it. And it wants you to compare both the crops across all 3 conditions. Do not only use the extreme one. Use all the 3 conditions, because using all the 3 is what gives you the patterns. Now, let's build the answer in exactly 3 layers. I'm going to walk you through it because this is a great template on how to structure any of the analyzed questions that you may find later. Layer 1, establish the baseline. Both crops perform similarly at 20 degrees and moderate light, which is 8.5 for beans and 9.1 for maize. This is your baseline. Why do you mention it? Because it shows the examiner you have noticed that C3 and C4 efficiency converge when the conditions are mild. Are you with me? The advantage of C4 is not present at mild conditions. You cannot notice it when the conditions are mild. It is conditional. It is not that every time C4 overlaps C3. No, it is at specific conditions. And you are showing the examiner that you know this difference. I'm saying you are going to show this nuance. It's very important to pick out these patterns and show them. Layer number 2. You are going to tell the examiner that at 30 degrees Celsius, the bean rate is going to fall to 6.1. Why? Because high temperatures raises the oxygenase activity of the enzyme Rubisco. Remember that one? Remember that Rubisco competitively has a high affinity for oxygen, and when temperatures increase, it tends to pick up oxygen instead of carbon dioxide. We are bringing it here now. So we are saying when the temperatures raise, oxygenase activity of the enzyme Rubisco increases. Now, this enzyme catalyzes carbon fixation in the Calvin cycle, but it is not perfect. It is sensitive. It can also bind to oxygen instead of carbon dioxide. You are showing the examiner that you know this. And this binding becomes more likely as temperatures rise. When Rubisco binds with oxygen instead of carbon dioxide, the plant undergoes photorespiration, which is a wasteful side reaction, which ends up consuming energy and releases previously fixed carbon without producing useful sugar. In the C3 pathway, Rubisco operates directly in the mesophyll cells and is fully exposed to atmospheric oxygen levels. So as temperature climbs, more and more of its activity gets diverted into phosphorespiration. This is why Beam's rate doesn't just plateau, it actively collapses. Okay? Now, the third layer. You are going to tell our teacher, you're going to write and say, maize, on the other hand, sustains 17.9 where it is unchanged. Remember, you have pointed out that beans collapsed. Now you are pointing out that maize actually remained the same. Why? And you're going to tell us the reason. Remember, we have what, why, and how. So we are stating the why. Because of the crans anatomy. Remember, they hinted on it in the scenario. So it is very relevant to bring it now. The very structural feature that we identified, in maize, carbon dioxide is first fixed in the mesophyll cells by an enzyme called PEP carboxylase, which has no oxygenase activity at all. It has no affinity for oxygen, meaning that no matter how high the temperatures increase, it cannot be distracted by oxygen the way Rubisco can. Now, when it combines with carbon dioxide, it produces a 4-carbon compound, which is exactly where the name C4 comes from. This 4-carbon compound, oxaloacetate, is going to be shunted into the bundle sheet cells, where it releases carbon dioxide at a high local concentration. Now, it is in this area where it releases this that we are going to have very high concentrations of carbon dioxide, where we are going to have Rubisco. This is going to remove the competition. Now, because by doing this, the C4 plants create an artificial environment that is highly concentrated with carbon dioxide. It means that oxygen cannot simply compete for the active site of Rubisco because there is a high concentration of carbon dioxide. Now, I want you to remember from characteristics of properties of enzymes that if we have a competitive inhibitor, increasing substrate concentration is also going to reduce the effectiveness of that inhibitor, because if it's a competitive inhibitor, it has to sit fast in the active site. If we have very many substrate molecules, for example, in this case, Rubisco, carbon dioxide, let's say oxygen is the inhibitor. If we have a lot of carbon dioxide, the chance of carbon dioxide sitting in the active site increases more than the chances of oxygen sitting in the active site. This means the C4 pathway is essentially built in a carbon dioxide pump that protects Rubisco from photorespiration. Now, let's ask ourselves, what makes this a great answer? Instead of just being a good or a correct answer, there are 4 points that I'm going to talk to you. Number one, it compares all the 3 conditions, not just the one that is dramatic at the end. It names the precise mechanisms where we are talking about Rubisco, oxygenase activity, what we call photorespiration. It does not just say C4 plants are efficient. No, it tells us how C4 plants are efficient. It tells us that these C4 plants have a certain leaf structure that makes them more efficient. The last point that we have talked about, which is talking about the crans anatomy, is very important because examiners are going to look for specific evidence used when you're describing your answers. And a sentence with a number in it reads as evidence. A sentence without numbers is like a guess. It's as if someone came across the correct answer by chance. But if you state figures that were given to you in the table, you are going to be more likely going to end up with answers that are biologically correct. Let's go to the next question. This question is saying, examine, and notice the question has also shifted focus. We are no longer just explaining the temperature collapse. We are examining how environment and leaf organization interact. That word interact is very important in this case. So our question is saying, examine how environment and leaf organization interact to influence productivity. This question wants 3 things. It wants you to explain how light intensity and temperature act together, not separately. It wants you to link the chloroplast distribution, that is mesophyll versus bundle sheath, to the biochemical division of labor between them in crans anatomy. And it wants you to show why this interaction matters for overall yield, not just the instantaneous rate that we measured in the lab. So let's build the answer. In maize, two chloroplast types aren't identical because we said in maize we have two types of chloroplasts. These chloroplasts that are found in maize are specialized for different purposes. We have the mesophyll, which are granular rich, meaning they have very many thylakoid membranes. And remember, it is in the grana where the light-dependent reactions happen. Now, this is where light is going to be converted into ATP and NADPH. Pay attention to that. The bundle-shaped chloroplasts, in contrast, are granular poor, meaning they are not built for light-dependent reactions. Instead, they are built for the light-independent reactions, in quotes, the Calvin cycle, where carbon is actually going to be fixed into sugar. Now, this is a physical division of labor between the two cell types. One cell type captures light energy, and a different cell type uses that energy to fix carbon. And critically, this division keeps carbon dioxide concentrated around Rubisco even as temperature rises, because the carbon dioxide releasing step is physically walled off inside the bundle sheet cell away from the atmospheric oxygen. Beans do not have such a separation. Every single mesophyll cell in the bean has to run both light reactions and the Calvin cycle in the same component. So as temperature rises, two things happen simultaneously, which make the situation worse. Number one, the stomata begin to partially close. I hope you remember that from coordination in plants, which is a plant's water conservation response to heat. And as a side effect, this reduces how much carbon dioxide can go into the cell by diffusion. With less carbon dioxide available and no concentrating mechanism to compensate, Rubisco becomes increasingly likely to bind the much more abundant oxygen instead. So you can see, it is not just temperature alone. It's not just light alone. It's an interaction of the two. High temperatures also trigger stomatoclosure. Stomatoclosure lowers internal carbon dioxide. Lower carbon dioxide combined with high temperatures is the condition under which Rubisco oxygenase activity dominates. Now, why does this matter for yield? It's not just for instant rate that we measured, because a growing season isn't one measurement. It is months of daily photosynthesis accumulating into biomass and grain. If maize sustains its photosynthetic rate day after day, while beans rate periodically collapses every time an afternoon gets hot, the difference is going to compound. Small daily advantages multiply over a whole season into a big difference in yield, which is exactly the real difference that farmers are going to notice as the outcome in our scenario. Now, ask yourself, why does this answer become a better answer? A weak answer would simply tell us what it said in the first question, talking about the plant anatomy, the bundle sheet cells, and the mesophyll cells. That's it. This answer instead is going to tell us the functional reason of the two chloroplast division, what each one specifically does, and why splitting labor under heat stress matters. Notice that if a weak student simply tells us the stating of the type of arrangement and stops there, an excellent student, which is what we are training you to be, is going to go ahead and tell us why this arrangement, why it works, why it eventually leads into good yield. Now, he's going to extend this single moment of data into a whole season and also tell us the consequence of this data expanded outside into a whole season. That's the difference between describing a structure and examining the structure in depth, which is exactly what this command word wants from us. Let's do the last task, which is talking about evaluation. This task says evaluate strategies to sustain crop yield under rising temperatures and water limitations. Evaluate strategies to sustain crop yield under rising temperature and water limitation. This question wants you to list at least two strategies that are grounded in the biology we have already established. It wants you to use that data used to justify each strategy, not just stating assertions. No, this is the part students forget. It wants a limitation or a trade-off acknowledged for each strategy, not just one. Each strategy must have a limitation stated or a trade-off. Okay? Now, let's go through the three strategies, because giving three instead of a minimum of two is a good habit when you have time. It shows that you have range. Strategy one. Our first strategy is shifting towards C4 staples in heat-prone season. Now, in this case here, the data clearly supports this, that maize sustains roughly 98% of its peak rate even when the temperatures are at 38 degrees Celsius, while beans collapse by almost 40%. This evidence is compelling, but there is a limitation. Cultural food preference limits how completely farmers can substitute one staple food for another. So you cannot simply tell an entire farming community to stop growing a crop that is central to their diet and their income. However, biologically, it is biologically the swap is sound to make. So when this tells us something about evaluation answers, it is important to tell us both the facts, allow the people to choose for themselves. That yes, this is a good strategy, but it has this limitation, it has this trade off. Our second strategy, targeted irrigation, timed specifically to flowering and grain field stages. Rather than irrigating uniformly across the whole season, why this specific timing? Because keeping stomata open during the plant's highest demand period prevents carbon dioxide starvation and will drive beans yields, will drive beans photorespiration spike in the first place. If you can keep internal carbon dioxide levels adequate precisely when the crop needs it the most, you are going to sidestep the entire mechanism that we discussed in task 1 and task 2. This is a more immediately actionable strategy than substituting full crops. But its limitation is resource availability, because the infrastructure for irrigation costs a lot of money and it requires water availability and access that not every small farm holder can actually be able to afford. This brings us to our third strategy. Breeding or agronomic practice approaches that improve beans' own heat tolerance. For example, instead of just planting any variety of beans, let us select for bean varieties with reduced rubisco oxygenase sensitivity, so the enzyme is simply less prone to that wasteful oxygen binding reaction even at high temperatures. This strategy has a real advantage than the first one doesn't have. It preserves the diversity. Rather than forcing communities towards a single staple crop, we are saying, no, let's keep our food, but let's choose better varieties. But it has a limitation, and that limitation is time. Breeding programs take years, sometimes decades, to produce a stable, improved variety. So this is not a strategy that you can deploy in this season or even the next season. Now, I want you to notice something. I want you to notice how I have arranged these strategies. They don't all operate on the same time scale. Strategy 1 is immediate. Stop planting beans, start planting maize. Strategy 2 is short-term to mid-term. Building the infrastructure for irrigation may take a year or two. Strategy 3 is long-term. It is a multi-year breeding program. Now, it is important for you to recognize that different strategies solve the problem on different time scales itself. This is also a piece of evaluation. You knowing that this particular strategy may not work now, but it might work later, is proof that you have evaluated it. Now, every strategy that we have used carries an explicit limitation. The strategies operate at different time scales. The whole answer connects the biology that we are actually talking about in the earlier tasks. And I want you to remember one thing from this task. Remember that an evaluate answer that only lists positive is by definition an incomplete evaluate answer. No matter how much excellent biology is in it, always force yourself to write the limitation. Okay? So let's have a very quick self-assessment checklist. So number 1, I identified the crop types C3 or C4 and the environmental challenge. This is the first step, and it should be the first thing you always do. Number 2, I spotted the hidden biological clues in the context introduction. Forgive the noise. I have some children that are playing physical education outside, but I know you can hear me clearly. Number 3, I read the data and identified the patterns before answering Not after, not before reading questions, not after reading questions. Before I read the questions, I identified the patterns. Number 4, I used specific values from the data to support every explanation. I didn't simply state vague statements, much higher, this is this. No, I stated specific points. So instead of saying much higher, I stated 17.9 versus 6.1. Number 5, I trust the biological mechanism, not just the named outcome. I'm not just saying C4 plants photosynthesize than C3 plants. No, I'm saying PEP carboxylase shuttle. I'm explaining the bundle sheet concentration. I'm showing how photorespiration is going to be suppressed. Number 6, I use precise vocabulary. I'm talking about guard cells, Calvin cycle, PEP carboxylase, exact words that are used in this biology because they are very important in the examiners awarding you marks. Number 7, my strategies are biologically justified. They are not just common sense. I'm not just saying use more water. That is common sense. But you telling me timing irrigations to flowering and grain fill to prevent carbon dioxide starvation during peak photosynthetic demand, that is biologically justified. Number 8, I address the limitations of my strategies and their trade-offs for all the strategies that I propose. This is very important because every time you evaluate and you forget this, your evaluation is not complete. Number 9, I connected at least the top 2 topics that are found in the assessment objective that I am answering. Remember, the strongest scenarios are going to deliberately go beyond one assessment objective. And finally, I matched my thinking to the level of the command word used. Now, my honest advice is when you are revising, when you don't have a paper, keep this checklist near you. Okay? And I want you to note every time you do a question, I want you to note and ask yourself, does my answer fit all the answers in the checklist? Check through very fast. And before you know it, it's going to be part of your head. You will remember it at the tip of your fingers. Now, let's bring this all together. Crop and climate, the hidden clues, the data pattern, the command words, the strategies, the 5-step chain, all these things here that you are saying, every scenario you meet from this point forward, whether it's about sorghum, whether it's about rice, millet, beans, greenhouse, tomato trial runs, exactly are going to follow the same chain. Identify what you are growing and the stress that it is undergoing. Find the clues the questions are quietly giving you. Read the pattern in the data before you read the tasks. Match your answers to the depth that is required of you, the command word that is given to you. And then, if the task asks for a strategy, always include the limitation alongside the benefit. I want to leave you with the same message I left with you at the end of assessment objective 1, because this, I think, is the most single useful thing in this entire course. The content is going to change between assessment objectives, but the way we think is not going to change. You have now built and practiced this exact skeleton twice, in assessment objective 1 and now in assessment objective 2. We did it for cells and respiration. Now we have done it for plant physiology. This skeleton is yours. I want you to get used to it. I want you to revise it, go through it, think on it. And as you think on it, apply it to assessment objective 3, which we are going to put on next. Animal systems and health survival, circulation, immunity, homeostasis. Apply this same type of thinking. Practice with this with your scenario workbook. And if you've not yet got this workbook, please send me a message. The workbooks are available. Some of these questions are coming from a big workbook that you can use to do as many questions as possible as you prepare for your exams. I've remained Educator Fresh. My name is Solomon. Thank you for watching. Please like and subscribe. And if this video has helped you, share with your neighbor, share with your friend, and I see you next time. Bye-bye.